Welcome back to MLT 240. We're going to continue our lecture for Unit 4. In this section, we're going to cover Mycobacterium. First thing that we're going to talk about is tuberculosis or TB. And here's just a little bit of history about tuberculosis. It was a disease also known as consumption or wasting disease or the White Plague. It affected humans for centuries until the mid-1800s. And people thought that tuberculosis or TB was hereditary. Since 1985, however, the number of new cases has increased by 14%. We can attribute this recent increase in TB cases into 4 factors. The HIV epidemic, the immigration from new countries where TB is more common than the U.S., the spread of TB in certain settings, and inadequate funding for TB control and other public health efforts. Let's talk about the Mycobacterium transmission. The transmission is person to person through the air. The droplet nuclei is 1 to 5 microns, so a droplet nuclei can remain suspended in the air for several hours. Not everyone who's exposed to an infectious TB patient becomes infected with Mycobacterium tuberculosis. The probability that TB will be transmitted depends on 3 factors: how contagious the TB patient is, in what kind of environment did the exposure occur, and how long did the exposure last. So here are some conditions that increase risk of TB infection, and I'm not going to read through all these. You can read through all of them on your own, but some include infection with HIV, the virus that causes AIDS, injection of illicit drugs, a recent TB infection, diabetes, prolonged therapy with corticosteroids, severe kidney disease, low body weight, and so on. Now let's talk about Mycobacterium pathogenesis. The infection begins if a droplet nuclei reaches the villi. The bacilli or rods multiply in the villi, and a small number may enter the bloodstream and spread throughout the body. Upper portions of the lungs, the kidneys, the brain, and the bone. The body's immune system usually intervenes, halting multiplication and preventing further spread. The ability of Mycobacterium tuberculosis to cause disease depends on the capacity to withstand intracellular digestion by macrophages. Remember what a macrophage is, which line it comes from. It comes from the monocyte line, which is the myeloid side. 10% of people who have TB infection will develop disease at some point. The remaining 90% will stay infected but free of the disease for the rest of their lives. Now we need to discuss the difference between TB infection and TB disease. Infection, you're going to have the tubercle rods or bacilli in the body. You're going to have the tuberculin skin test reaction, which is usually going to be positive. The chest x-ray is usually going to be normal. Sputum smears and cultures are going to be negative. You're usually not going to have symptoms in infection patients. They're not infectious, so they're not going to spread to others. And so it's not a true TB case. Okay, but the disease, on the other hand, which means it's made it to the lungs, you've got the rods in the body, you have the tuberculin skin test that is usually positive, you have the chest x-ray that is usually abnormal, sputum smears and cultures are positive. There's going to be symptoms in our patients such as cough, fever, and weight loss. Often infectious before treatment, so you can spread the disease to other people or other patients, and this is a true case of TB. Mycobacterium treatment for TB is usually a multi-month, multi-drug regime, often including the following 3 drugs. Rifampin is usually the first line in conjunction with other antibiotics. Isoniazid is also used, or INH, in streptomycin, and those are both antibiotics. Some general characteristics when it comes to Mycobacterium. They're slender, slightly curved or straight, or rod-shaped organisms. They're non-motile, no spores are formed. Cell walls have extremely high lipid content, causing microbacterial cells to resist staining with traditional stains. They usually resist decolorization with acid alcohol or ethanol, and this is referred to as acid fastness. They're strictly aerobic, enhanced with CO2. They usually grow slowly and can take up to 6 weeks. When collecting, you want to collect a first of the morning specimen. When culturing, the appropriate media can be the Lowenstein-Jensen or the Mehlbrook is common, cause the growth is better, but it's faster in a liquid media as far as growth. And the preferred carbon source for Mycobacterium is glycerol. These are Mycobacterium tuberculosis complexes. They can also be referred to as MTC or MTBC. These are genetically related groups of Mycobacterium species that can cause tuberculosis in humans and animals. And again, these are all fall in that Mycobacterium tuberculosis complex. So these are all MTCs. Okay. We can also have non-tuberculosis Mycobacteria, or NTMs. These are found in the soil and water, and sometimes they're also referred to as MOT, or MOTT, Mycobacterium, other than tuberculosis. And these are usually opportunistic pathogens in immunosuppressed patients. These are not transmittable from person to person. And some of the species that fall within this category are included down here. For Mycobacterium labidae, we have the acid-fast stain and the Kenyan stain. Let's first talk about the acid-fast stain. The primary stain is the carbofusion stain. It binds with the mycotic acid in the cell walls of the Mycobacteria and stains red. It is retained after the decolorizing step with acid alcohol. The counterstain does not penetrate the Mycobacteria or affect the primary stain, and the stain's background material is an acid-fast blue. For the Kenyan stain, we're going to cover the smear with the carbofusion stain and for 5 minutes and wash with water. You're going to decolorize with the acid alcohol until it runs clear. Then you're going to rinse with water. Then you're going to flood your stain or smear with methylene blue for 1 minute, and then you're going to rinse. And we're going to look at an example of both of these stains here in just a second. Here is the Mycobacterium tuberculosis with the acid-fast stain. The red is the bacteria, and the background is blue. This is the Kenyan stain. The red is due to the carbofusion, and the blue is due to the methylene blue. Another stain is the Ziehl-Neelsen stain. You cover the smear with filter paper. You add the carbofusion to the filter paper. You heat the smear gently until it steams. Then you stain for 5 minutes without heat, and then you proceed on to the Kenyan stain. Here is an image of the Ziehl-Neelsen stain. The red is the acid-fast bacteria. The blue is the non-acid-fast bacteria. The procedure for the Ziehl-Neelsen is on the next slide as far as a picture diagram, and we're going to look at that in just a second. So here's the Ziehl-Neelsen staining procedure. You have the application of the primary stain, the carbofusion, then the application of the heat, then the application of the acid alcohol or the decolorizer, and then an application of the methylene blue, which would be part of the Kenyan procedure. Specimen concentration method. So what do you need to know for collecting specimens? Our specimen for Mycobacterium most likely is going to be sputum. We need to liquefy the sample and break up the mucus through digestion of proteinaceous material. So proteinaceous just meaning material containing proteins. And we're going to use NaLC to do that. We also want to kill non-microbacteria and decontaminate, and to do that we're going to use NaOH. So we've said already that our specimen is going to be sputum. We want to use first in the morning, 1 a.m. specimen. You can also use a gastric specimen, a bronchial wash, or a transtracheal aspirate. But usually our preferred specimen and our easiest specimen to get is our sputum specimen. The reagents, again, we've just mentioned NaOH. It kills normal oropharyngeal flora or many non-microbacteria, and then our NaLC, which breaks down mucus basically. So it just helps liquefy and makes it thinner. We want to use a media that's aerobic and it's enhanced with CO2. Again, we've said these are slow growers. It can take anywhere from 2 to 6 weeks for a pathogenic microbacterium to grow. The media, we can use an egg-based media like Lowenstein Jensen or LJ agar. We could also use the American Thoracic Society or ATS agar. We could use 3 different types of serum agar-based. We could use the Melbrook 7H110 or 7H11. There's a couple of varieties. They must be refrigerated and incubated in the dark. Light causes the release of formaldehyde, which is toxic to Mycobacterium, so that's why we like to leave it in the dark. Liquid tends to be a little bit better. It helps microbacteria grow more rapidly, so it might be better to use a broth media. We can use the Bactec broth, or we can use the liquid Melbrook 7H9. For colony morphology, we can have anywhere from smooth or rough appearance, but as far as the rods, they can be corded looking. They can be in curved strands of rods or bacilli. They fall into non-chromogen or a non-chromogen category as far as color, or they're very light in pigment. And they also can fall in a Runyon category group. Runyon is a classification system of non-tuberculosis microbacterium based on growth, pigment, how pigment will change when exposed to light, and we'll discuss these categories a little bit later on. Also, you can use the growth rate, whether it's slow or rapid, to help ID, and also temperatures. Does it grow better at a lower temperature, 22 to 35 degrees Celsius, or a higher temperature of 33 to 39 degrees Celsius, as ways to help identify. For Mycobacterium tuberculosis, the colony is slow-growing colonies with a dry, rough appearance. The color is anywhere from buff-colored to colorless, and it grows slowly at temperatures in the range of 35 to 37 degrees Celsius. Biochemical test, the catalase is going to be positive. The antibiotic test for INH is going to be resistant. The niacin is going to be positive. The nitrate reduction is going to be positive. Tween 80 hydrolysis is positive after 10 days. And you're going to see cording if you're looking at the bacilli under the scope. Of course, we know Mycobacterium tuberculosis can cause TB. It can cause pulmonary, extrapulmonary problems. Transmission for Mycobacterium tuberculosis is through contact. It can be airborne. It can even be transmitted through milk. Of course, treatment. is through antibiotics. To detect Mycobacterium tuberculosis, we do a skin test. We can do a skin test called a PPD. This is a purified protein derivative. It's administered intradermally, and what we do is it contains a killed filtered organism, and you use a syringe and needle, and it's injected just right underneath the skin. If there's a greater than 5 millimeter interduration, that means it's a positive test, so it's positive for Mycobacterium tuberculosis. The older skin test is known as the time test. The old tuberculin was in glycerol, and it was heated up to kill. And you had just a small little prick of the skin on the forearm, and the killed organism was introduced to the skin. If you had a greater than 2 millimeter in duration, that was a positive skin test for Mycobacterium tuberculosis. For morphology, let's talk about micro versus macro. For micro, Mycobacillary to long, narrow rods, and it's going to be beaded and non-spore forming. Micro, how the media is going to look on the plates, what you can see with the naked eye. It's going to be dry, buff to yellow cauliflower colonies on the Lowenstein-Jensen media. It's strict aerobes, and of course it's going to be enhanced growth with CO2 at 2 to 3 weeks. So here are those cauliflower looking colonies of Mycobacterium tuberculosis. Now let's switch gears a little bit and talk about Mycobacterium leprae. It's a causative agent of leprosy or Hansen's disease, infection of the skin, mucous membranes, peripheral nerves. There's two forms. There's the tuberculoid skin lesions with nerve involvement producing areas of anesthesia or loss of sensation, and spontaneous recovery often occurs with this form. And there's leprematosis, which is progressive, and it's usually malignant or cancerous, and it's life-threatening. Despite the reputation, it's not easily transmitted or transmittable, and it's rare in the U.S. It can be transmitted by direct contact and through inhalation of aerosols from the skin lesions. Mycobacterium leprae cannot be cultured on artificial media. And to get around that, it used to be cultured on the foot pads of mice. The optimal growth temperature is 30 degrees Celsius, and the diagnosis is usually through skin biopsies. Here's a couple of images of patients that are suffering from Mycobacterium leprae. Alright, now let's talk about Mycobacterium avium complex, or MAC. This is a complex made up of M. avum and M. intracellular. It's a common environmental saprophyte recovered from soil, water, household dust, and other environmental sources. Increased numbers found in marine sources. So this is also known as environmental TB. It produces non-tubercular infections. It does cause pulmonary infections, and it presents like TB, and it is the most common symptomatic bacterial infection in AIDS patients. As far as lab ID, because the two species are so similar, most labs will not distinguish between the two, but will just report out it as a MAC, or a Mycobacterium avum complex. It grows slowly on primary growth media. Optimal temperature is 37 degrees. Produces a smooth, non-pigmented colony that become yellow with age. These are mostly biochemically inactive, except for producing heat-stable catalase. Next is Mycobacterium cancericea. It's the second to MAC as the cause of non-tuberculosis Mycobacterium for lung disease. It's not transmittable person to person. It's the most common manifestation of pulmonary disease resembling TB involved in the upper lobes. It usually causes cavities to form or holes in the lungs, and it also causes scarring in the lungs. Other infections reported are skin infections, lymphadenitis, which is swollen lymph nodes, and soft tissue infections. Infections rarely are seen in immunocompetent patients or individuals. Lab ID includes this being a slow grower. It appears in long rods with distinct cross-banding. Optimal growth temperature is 37 degrees, and colonies appear smooth to rough with wavy edges and dark centers when grown on the Melbrook 7H10. The nitrate and the catalase test will be positive. For photochromogenic, the colonies form a pigment when exposed to light, but are non-pigmented in the dark. Next is the Mycobacterium fortunatum group. It's a group that contains 10 species. These are common in the environment and isolated from soil, water, and dust. They're implicated frequently in infections of the skin and soft tissues, including localized infections and abscesses at puncture sites. Associated with long-term use of IVs, peritoneal catheters, injection sites, and surgical rooms. These are fast growers after 3 to 5 days at 37 degrees Celsius. Colonies appear rough or smooth, non-pigmented, and they are creamy white or buff in color. And the nitrate will be positive. Here is an example of an M. fortunatum infection on a finger. A couple other species of Mycobacterium we want to mention is Mycobacteria marinum. This is a marine organism. The infection is also known as swimming pool granuloma. Nodules develop in a patient, especially with a history of cleaning fish tanks or exposed to salt water or non-chlorinated swimming pools. Next is Mycobacterium gordoni. This is a non-tuberculosis Mycobacterium. The natural reservoir for it is water. It's natural and tap water. It's found in both, and also in soil, and it's often referred to as tap water bacillus. It also has been recovered from pools and unpasteurized milk. So now let's look at some different classifications of our Mycobacterium species. So this one is pretty simple. You have your Mycobacterium up here as an overall category. Then you have your Mycobacterium tuberculosis complex over here. And then you have certain species that fall underneath that category that can cause tuberculosis, either in humans or animals. Then you have this non-tuberculosis category here, or your MOT. Remember, our MOT stands for Mycobacterium other than tuberculosis. NTM stands for non-tuberculosis Mycobacterium. And the Runyon 1 through 4 categories are also going to fall within this same non-tuberculosis category. And we're going to talk about this category a little bit more here in just a minute. But over here to the far right side, you have Mycobacterium leprae kind of hanging out by himself. So let's talk about these Runyon groups, and these are based on photoreactivity. And how was these groups formed? In 1959, there was a botanist named Earl Runyon that put these NTM Mycobacterium into 4 categories based on photoreactivity, or the production of pigment in the presence of light or dark. So he's got 4 different groups. He's got group 1, which is slow growing photochromogens, which produce pigment when grown and exposed to light, no color change in the dark. He's got group 2, which are slow growing Scotochromogens, which produce orange to yellow pigment when grown in dark or light. He's got group 3, which are slow-growing non-chromogens that do not have pigmented growth, whether grown in the dark or light. And then there's group 4, which are rapid growers, which are fast growers within 3 to 5 days. So let's look at a chart on the next slide of what some of these, or where some of these different species of Mycobacterium fall. So here's the running classification as a chart. You have your 4 different groups, and some of the different species that we talked about in this lecture are placed in different categories within this table. You have group 1, which is your photochromogens, which produce that yellow pigment, and these are your slow growers. We've got a couple different species that we talked about that fall in that group 1. I don't believe we talked about any that fall within group 2. Group 3, we've got at least one species there that we talked about. And these do not produce any pigment. These are non-chromogens. And then you have your rapid growers. We've got one down here that we've talked about that grow rapidly at 3 to 5 days. So this is your Runyon classification, and how that works. It's based on pigmentation and photoreactivity of those species. When working with Mycobacterium specimens, you have to be careful, so we need to touch on some safety precautions. At a minimum, you should use a N95 respirator. You should work with samples under a level 2 biosafety hood and use appropriate PPE when handling cultures and samples. Patients are usually isolated in rooms with negative airflow to prevent droplets and aerosolization. Annual fit testing for respirators for all staff in patient contact or staff handling samples of Mycobacterium are required, and also the use of proper disinfectants are required. Even patients that are just suspected of having TB are usually placed in isolation. This concludes the Mycobacterium portion of the lecture. I will see you over in the next section.